This lecture is Muscular System, Structure, and Function. These are your learning objectives. Skeletal muscle function, of course, primarily is movement. And it works with bones in a way that the bones and muscles interact as levers. There's 4 components to a lever: a rigid rod or bar, and in that case this is the bone; a fulcrum or pivot on which the bar turns, and this would be the joint; an object moved against resistance, and this can be anything. This can be just lifting your hand against gravity, or picking something up, or walking across the floor. You're moving against resistance. And a force that supplies the energy, and this is where the muscle comes in, to supply the force to move the body. There are 3 different kinds of levers. The first is a first-class lever, and in this case you have the fulcrum in the middle. So force is applied to one end, resistance is applied to the other, and the fulcrum is in the middle, and they've given you kind of a real-life example of this, like a pair of scissors. So fulcrum, force, resistance. First-class lever, fulcrum in the middle. In the second-class lever, you have the resistance in the middle, the fulcrum and force on either end. Like a wheelbarrow, fulcrum here, lifting the handles would be the force, the resistance, what you're carrying, is in the middle. In a third-class lever, you have the fulcrum on one end, the force in the middle, and the resistance on the other end. So third-class, force is in the middle, like a pair of tweezers. Fulcrum on one end, force in the middle, moving against resistance. In your body, you have lots of first-class and third-class levers. You don't really have many second-class levers. So let's look at this one. This is flexion of the arm at the elbow. The primary muscle that is contracting here, the prime mover, is the biceps muscle, pulling the arm up against the resistance. Here they have this person lifting 10 kilograms. So the movement is this way, you're flexing, and you have force, fulcrum, resistance, although that is not exactly the correct order, because look at where the force is attached to the radius. Okay, the insertion of this muscle, the prime mover, is right here on the radius. So fulcrum, force, resistance, making this a third-class lever. If you're doing the opposite movement, if you're extending against resistance, then the triceps is the prime mover, the biceps is relaxed, and you have force, fulcrum, resistance. Fulcrum in the middle is a first-class lever. A little bit more on these terms about muscles. Okay, the origin of the muscle is the attachment of the muscle to a relatively immovable or fixed part. So here's the biceps brachii. Biceps because there are 2 heads, brachii in the brachial region of the body. And you can see that the tendons of the long and short head of the biceps are attached to the coracoid process up here on the scapula. Sure, you can move your scapula, but it's relatively immobile compared to where the insertion is, which is the attachment to the movable part on the other side of a joint. So here's the joint, here's the insertion on the radius, and this is the part that moves when this muscle contracts. The head of the muscle is simply the part nearest to the origin. So these are the heads of the muscle connected to these tendons. Muscle interactions. Which muscles are involved in flexion at the elbow? So we already talked about flexing the elbow involves the contraction of the biceps brachii. That is the prime mover or the agonist. So the muscle that is primarily responsible for making that movement, the largest contraction, is going to be your prime mover. It probably has some other muscles involved that are working with it, and those are synergists. In this case, the synergist for flexing at the elbow would be the brachialis muscle. The antagonist is the muscle that has the opposite effect of the prime mover. So when you're flexing at your elbow, your bicep is contracting. The triceps muscle on the back of the arm is relaxing. The triceps is antagonist to the bicep. If you are extending the elbow instead of flexing it, the tricep is contracting, the bicep is relaxing, and in that case the tricep would be the prime mover and the bicep would be the antagonist. So whether or not a muscle is a mover or a synergist or an antagonist really depends on the type of movement that you're making. So we're going to get into now detailed description of the skeletal muscle structure. And you start by talking about the layer of connective tissue called fascia that surrounds all of the skeletal muscles. Every skeletal muscle is covered by a dense connective tissue called fascia. The fascia at the ends of the skeletal muscles form tendons which connect the muscle to bones. So you can see lots of tendons here connecting these quadriceps down to the patella or down to the bones in the hand, to the elbow, okay, all to all of these joints. They also can form aponeuroses, which is a fibrous sheet, and that connects muscle to muscle. So here in the abdominal wall, all of these individual abdominal muscles are connected to the other abdominal muscles by aponeuroses or fibrous sheets. So this is the detailed organization of skeletal muscle. It is very organized, but there are lots of parts, and you kind of have to really think about the terminology as you're going through this. So we'll go through it bit by bit and level by level. So we've already talked about the fascia. Fascia covers the whole muscle and it forms the tendons and the attachments to the bone. You have layers of connective tissue that enclose and separate all of the different elements of the muscle, starting with the fascia. And then just beneath it is a layer of connective tissue called the epimysium. Okay, so epi means above or on top of, around. And so that is the outermost layer of connective tissue underneath the fascia, next to the muscle, mysium. The perimysium extends into the muscle and separates the muscle into separate fascicles. Okay, so here's another term. Muscle gets separated into fascicles by the perimysium. Perimysium separates the muscle fascicles. And now they've extended one fascicle out. Okay, so you can see you've got blood vessels here and a motor neuron. And here's the fascicle. And in between this one, we have separated the fascicle now into all of these little individual muscle fibers. And that is done by the endomysium. So the tissue that extends into the fascicles and separates into fibers is endomysium. Fascicles, endomysium, muscle cells, and fibers. And then the muscle fibers can be further broken down into myofibrils. Okay, so this is one muscle fiber. These are individual muscle fibrils. This is one muscle fibril, which is separated into thick and thin filaments. And we will go through all of these in much more detail. But generally, this whole muscle, the whole thing, covered by the fascia is called a single compartment. And you'll hear the word compartment when you talk about compartment syndrome, which is injury that causes blood from an internal hemorrhage to accumulate inside a compartment. So if the compartment stays closed, if the fascia is not injured, but the muscle is injured, and blood is accumulating inside that compartment, you have huge pressure increases inside the compartment. That rising pressure interferes with normal, healthy blood flow to the region. So you're increasing the pressure in the compartment, and the rest of the muscle that needs the blood is not getting the blood that it needs because of the pressure. So that reduces the supply of oxygen and nutrients to the muscle tissue. And when tissue doesn't get oxygen and nutrients, it begins to die. Dying muscle tissue in your body is a really, really bad thing. So physicians will do a procedure called a fasciotomy, where they literally just slice right through the fascia to relieve the pressure and restore the circulation to that compartment. Okay, now I want to look at the fascicle up close. So I've included these images up at the top corner with a red box around it so you can kind of orient yourself to what level we're at. So again, the whole muscle separated into fascicles, and now we're looking at a single fascicle. So the single fascicle is surrounded by perimysium, and endomysium separates the individual muscle fibers within. So one of these little circles is an individual muscle fiber or an individual muscle cell. A muscle fiber and a muscle cell are the same thing. So the whole thing is a fascicle, perimysium surrounds it, endomysium surrounds the individual muscle fibers or a single muscle cell, and then the muscle fibers are further separated into myofibrils. So here's more about the skeletal muscle fiber. Each skeletal muscle fiber is a single muscle cell that is multinucleated. So there's multiple nuclei in a single muscle cell. And they have some funny names for things. So the muscle cell membrane, instead of being a plasma membrane, is a sarcolemma. Okay, so sometimes you call regular cell membranes plasmalemma. This is going to be a sarcolemma. The muscle cell cytoplasm is the sarcoplasm. The sarcoplasm is full of mitochondria because muscle cells need a lot of energy, and it's also full of myofibrils. Sarcoplasm contains abundant myofibrils. The myofibrils are the contractile proteins in the muscle cell. I'm going to talk more about them on the next slide. I also want to point out the sarcoplasmic reticulum, which is the muscle cell's corresponding part to the endoplasmic reticulum, and its role is to store calcium for muscle contraction. So myofibrils are the protein organization that causes contraction. So here's a single, again, this is a single muscle cell, and within that single muscle cell there are multiple myofibrils. The myofibrils are made of thick myosin in the pink here and thin actin in the purple filaments. Okay, so these are protein filaments that actually are the contractile proteins in the muscle. They're organized into something called a sarcomere, which goes from Z line to Z line, and that sarcomere can shorten. And when the sarcomere shortens, the muscle is contracting. So you can see all of these sarcomeres organized end on end in the myofibrils, in the individual muscle cells. They all shorten at the same time, and you have the whole muscle shortening and the contraction occurring. More on that sarcomere. It has quite a few parts that are important, okay? First, we'll start by looking at the Z line. So a sarcomere goes technically from Z line to Z line, but they're all lined up end on end to each other. But they go from Z line to Z line, so that would be a single sarcomere. The I band is the part that surrounds the Z line that consists only of light actin filaments. So light purple, okay? Those are the thin filaments, the actin filaments, and they surround the Z line. So one sarcomere is going to have a little I band on one side and a little I band on the other. But you can see when they're lined up end on end, the I band extends completely across the Z line. The A band is the dark band. So this is the region of overlapping filaments and myosin filaments only. So here's the overlap, right? Because you can see there's actin and myosin here in these very dark parts, and there's myosin only in the middle, and that whole thing is called the A band. M is the midline, and that anchors the myosin. So the middle of the sarcomere also anchors the thick myosin filaments. And then there's the H zone. The H zone is the area where there's only thick filaments. So it is within the A band immediately surrounding the midline. Finally, there's a protein called titin, which holds the myosin to the Z lines, just a thin little protein, just so they don't flop around, I guess. Okay, so here is the whole sarcomere. And one way to remember this is to use the acronym MHAZI, MHAZI. The M line here is in, the M line is in the H zone, which is in the A band, and the Z line is in the I band. MHAZI. The M line is in the H zone, which is in the A band. The Z line is in the I band. If you take even a closer look, this is the myosin filament, okay, the thick filament with the myosin molecules. The M line would be right here down the middle, okay, and you can see the overlapping thin filaments. Now the thin filament is not just actin, because face it, that would be way too easy. It's got to have a few things in it. Actin is complexed with troponin and tropomyosin. So the troponin is the protein right here, and the tropomyosin is this curvy dark purple thing that covers up the little actin protein that look like beads inside. So these are the actin, the beads. The tropomyosin is the curvy coil, and the troponin is this other protein right here. And these become really important when we talk about muscle contraction, because the troponin and tropomyosin have to move in order to expose the actin and allow the myosin to bind, and the muscle to contract. And I'll explain that in much greater detail in the next lecture. A few more structures that are important. I've already mentioned the sarcoplasmic reticulum. Its job in the muscle cell is to store calcium. We know that calcium regulation is really, really important. We talked about it when we talked about bones. And one of the reasons it's really important is because it's necessary for muscles to contract. Okay, so the sarcoplasmic reticulum stores calcium. You have transverse tubules, another important structure, especially important in this triad area. So the transverse tubules run through the center of the edges of the sarcoplasmic reticulum, also called the cisternae. So it's like a triad, the transverse tubule in the middle and the two cisternae of the sarcoplasmic reticulum abutting it. So in this triad region, when a signal for the muscle to contract is sent into the muscle, it travels the transverse tubules. So that's called signal propagation, sending the signal. The transverse tubules, or T-tubules, send the signal through the myofibrils, and every place that they abut these cisternae in these triad regions, they release calcium from the sarcoplasmic reticulum. Okay, so it's the signal going through the T-tubule that makes the sarcoplasmic reticulum release calcium, and that is what actually causes the muscle to contract, which we'll see again in the next lecture. Okay, just briefly, we're going to talk about smooth muscle and cardiac muscle. So what makes smooth muscle smooth? Well, it's smooth because it's not striated. Cardiac muscle has striations, and so does skeletal muscle. And the striations are caused by the organized pattern of the myofibrils and the thick and thin filaments. Smooth muscle doesn't have any of that. There are two types of smooth muscle, multi-unit smooth muscle and visceral smooth muscle, distinguished by how they contract. So I'll talk about that in the next lecture. And then there's smooth versus skeletal muscle structure. So this is a good comparison slide. Smooth muscle, I've already said, has no striations. And these cells are short and they have tapered ends, whereas in skeletal muscle, the cells are long, arranged end to end in their sarcomere pattern. They have centrally located single nuclei, whereas skeletal muscle cells are multinucleated. Their filaments are thin and randomly distributed, whereas skeletal muscle has organized, dense actin and myosin filaments. There are no transverse tubules, and the sarcoplasmic reticulum is not very well developed, where in skeletal muscle, these two structures are absolutely essential. They're well developed and important for sending the signal to contract. Cardiac muscle, on first glance, looks a little more like skeletal muscle, except that like smooth muscle, it only has a single nuclei for each cell, and it has these features that make it really distinct from either smooth muscle or skeleton muscle, these intercalated discs. The intercalated discs are the ends of the cardiac muscle cells, but they form really nice complex junctions between the cells. And the intercalated discs allow the force of the contraction to spread really rapidly between cells so that the whole heart can contract together as it's supposed to. This tissue is self-exciting, which means it stimulates itself, and then it will contract all together in something called a syncytium. It's also a rhythmic contraction. You know, your heart rate is very regular, rhythmic beat. Okay, so here are the key points of structure and function for skeletal, smooth, and cardiac muscle to compare and contrast. And that marks the end of this lecture. Thank you.